are. So for here, we can see that microplastics, which comes from plastics, they influence or impact the brain, muscle, kidney, gills, stomach, intestines, and ovary. But most of all is the eventual effect it has on locomotion, physiological impact, reproductivity, growth, and development of the fish that we are trying to catch through wild catch or even aquaculture. So it is going to affect the fishery industry and aquaculture industry. Next, this slide is about a solution, an option where PHA, a bioplastic, might partially substitute the plastics to lighten the effects of plastics towards biological systems. So this is an example of the degradation of PHA, whereby PHA can be degraded under the presence of oxygen, microbial activity, and its physical chemical properties, which can vary from one to the other type of PHA, since PHA is a family of bioplastics with many species or individual types. So they are biodeteriorated into fragmented plastics by these environmental conditions that can speed up or slow down the process. And then they are further biofragmented into oligomers and monomers or microplastics that can be assimilated into water and carbon dioxide. But this is the case for polyhydroxyalkanoate bioplastics. So in the next slide, let me begin by sharing the applications of PHAs. So from this slide, we can see that this material is very versatile. For example, they have applications in the major industries or sectors of industrial, agricultural, and medical. The main role of PHA is actually in the medical sector, especially as sutures, which is shown in this figure here, and bone tissue engineering and artificial heart valve and blood vessel. However, they are also increasingly developed for food packaging in industrial and agricultural net and encapsulation in agricultural industries. So next, this is the biosynthesis pathways of PHA. The main pathway involved are fatty acid de novo synthesis pathway and fatty acid degradation beta oxidation pathway. And the main genes, although we can see that there are about 5 PHA synthesizing genes here, but the core genes that almost all PHA producers have in their DNA is the FAA, FAB, and FAB-C gene, whereas the most important gene is FAC. Why? Because this is the polymerizing gene which polymerizes monomers into polymers that eventually are called PHA. So why is it so important? Also because FAC gene properties have substrate specificity, meaning that they will either convert sugar, oil, or waste into PHA, but not all of this because they have selectivity. They select their substrate. And so, because of that, FAC can influence the carbon chain length, monomer composition, and eventually the physical properties of PHA. So the good news is that this makes PHA a very versatile material that can be adjusted into different physical chemical properties. But the downside here is that we will need to research more on and discover more types of FAC that can convert them into even broader spectrum of PHA with different physical properties. So the common microbes involved are Pseudomonas bacteria, Bacillus bacteria, and Cupreavidus bacteria. These are the most common microorganisms involved. Yes, other bacteria species or genus can produce PHA as well as fungi and microalgae. However, the most flexible and ease in terms of efficiency are bacteria and only certain species in the genus, meaning if not all species in Pseudomonas genus can produce PHA, only some species in that genus. So next, in terms of the production of PHAs, it generally involves microbial fermentation and some downstream processing. So over here is a figure showing the detailed method of producing PHA, which is the most recognized method of producing PHA. However, the current advancement in this area in terms of production is to try using and discovering new feedstocks or substrates that can be used to replace sugar and oil. Like what is mentioned in the previous slide, we can produce PHA from sugar and oil by feeding them to bacteria, but that is known as not as sustainable and renewable because it might threaten the human food source when we use sugar and oil. Therefore, the current advancement is to use waste oils, soy molasses, crude glycerol, and starch that are from potato and cassava waste as a feedstock or substrate in producing PHA. So in the next slide is something about PHA-based composites. For now, it is still a very young field in terms of pure PHA products and composite PHA products. There are even some new research on producing smart PHA composite material. However, purely in PHA-based composites, they are known to improve their properties in terms of biodegradability, especially, and then mechanical and chemical properties. However, we should also remember that the physical chemical properties of PHA can be also influenced by the gene that or enzyme that we use. And over here are some common fillers used to produce these composites, which are starch, fibers, and other bioplastics such as polylactic acid. The grass fibers are usually used, which are grass such as jute and miscanthus, also known as silver grass, shown in these two images here. In the next slide, this is trying to share the innovations of PHA, whereby we will source them from the environment and then we will screen for beneficial bacteria that have the ability to produce PHA, and then we will go through these several processes such as gene mining and natural product extraction and synthesis. So the current PHA research is metabolic engineering, fermentation, optimization, and mechanical property enhancement according to Lau 2017 and 2014 as well as Al-Saadi. So next, the PHA has several environmental and economic considerations, although it is known as a biodegradable plastic. So over here, we can see that the usual way of producing a manufacturing line is a linear process from cradle to grave, meaning from raw material and to a product which cannot have any more value added. However, by adding PHA, using PHA into this process, we can create a circular economy concept, a cradle-to-cradle concept, which is circular, whereby after the product is formed, we can use the waste formed from the product as well as the byproducts from the manufacturing process to feed it to microorganisms and ferment it to produce PHA, which is then formed into different applications such as shown previously in the three major industrial sectors. So
Okay,
for
can we have
future,
last 2 minutes for you
yes,
to discuss?
Thank you. Sorry. So, for future directions and conclusions, we have to focus on the cost and yield of PHA production to optimize the processes, such as using simulation modeling and then circular economy to integrate it into the PHA manufacturing process. Next is the use of feedstock and substrate that are sustainable, such as oleochemical and agricultural waste. And lastly, the implementation or enforcement of governmental frameworks for proper management strategies and policies. Okay, thank you. These are my references. And do you have any questions?
So thank you so much. Using green routes. When we talk about the environmental remediation, it is a need of the hour. Why it is the need of the hour and why the green routes are required, this will be the main theme of my presentation. The remediation involves all type of remediations in terms of pollution, pollutants, and all sort of unwanted or harmful products or the leachates which are added to the environment on daily basis, and they lead to a lot of degradation of the not only the soil, air, and all the resources of the earth. And therefore we need more and more green routes. When I talk about green routes, it means those alternate sources of energy which we can use and utilize or harness these resources for making more sustainable environment. So proceeding further, just a minute. Is it visible? Another slide is visible?
Yes, ma'am. Could you
Okay.
please go to the
So
presentation
can you
mode?
imagine life without fuel? No, we cannot imagine life without fuel because we need it for the day-to-day transport. So what is
Sorry,
sustainable?
ma'am. Ma'am,
Yeah.
sorry to disturb you. Could you
Yeah.
please go to the presentation mode, like full screen?
Okay, sir.
Yeah.
Okay, it's fine, sir.
Okay, please go ahead.
Okay. So what do you understand by promoting low sustainable environmental remediation using green routes? We are talking here about the promoting low carbon transport in India. India is currently the fourth largest producer of greenhouse gases in the world, with its transport sector being the second largest contributor of carbon dioxide emissions, which is contributing to the carbon footprints on daily basis. There's need to reduce transport emissions through the adoption of sustainability approaches, that is switching to more and more biofuels or alternate sources of energy. Promoting low carbon transport in India was a major transport initiative of United Nations Environmental Programme, and the project built a network of expertise in transport planning, safety, social inclusivity, and air pollution and climate change. So this promoting low carbon transport in India was an initiative taken by the United Nations Environment Programme. Now, what does this policy include? The low carbon transport system policy is a system demonstrating the usage of alternating fuels or alternative sources of fuels, which can be harnessed or reduced, and they can be very important for reducing emissions while helping to meet the increasing demand for global transportation. It also ensures for production of low carbon transport fuels in sustainable manner with low or negative footprints on our ecosystem. Another very important feature of this policy is it provides for economically viable infrastructure and operation that offers safe and secure access for both persons and the goods, whilst reducing short and long negative impacts on local and global environments. And what are the key benefits of this policies? These are listed. Some of the key benefits are reducing carbon dioxide emissions, balancing the ecosystem components. Another very important advantage is reduce the fuel consumption for transport, promoting sustainable transport, and cheap cost of transportation is also involved because everybody can access these fuels. Another very important feature is minimizing the climate change impacts. Now, what are various steps which needs to be taken
Excuse
for
me, ma'am. Excuse
switching
me,
to
ma'am.
this type of policy?
Excuse me, ma'am.
Yes, sir.
Ma'am, your slides are not changing. I mean, we are still on third slide.
You're not able to see? Is it
We are
changing
able to.
now?
Yeah, no, ma'am.
Okay, just a minute, sir. I'm just resharing. I think there's
Sure,
some.
sure. Just reshare, ma'am. Yeah.
Yeah.
Ma'am, instead of sharing a single tab, please share the entire screen. Then it would be
Okay,
like,
entire screen. Okay,
yes,
yes.
ma'am. Yes.
So I have shared. Is it
Yes,
visible now?
yes, ma'am. Could you please go to
Okay.
the presentation?
And I'm just going to change the slide. Is it visible?
Yes, ma'am. Now it is.
Okay, fine, fine. Sir.
Can you please click on this hide button?
Yeah,
Yeah, sure. Yeah, hide
yeah.
it. Thank you.
Okay.
Please go
So,
ahead. Sorry.
yes, sir. So, what are the steps involved towards switching to this type of policy? The UN Climate Change Conference in 2016, which was held in Morocco, provided for the global climate action agenda on transport and tackling emissions from the transport in both possible and cost-effective manner. In this direction, the GCAA, that is General Civil Aviation Authority Transport Initiative, has started various programs such as Mobilize Your City initiative to tackle the problems of transport emissions, and various initiatives like Green Urban Transport Schemes, New Metro Rail Policy, Revision of Metro Acts, and standardization and indigenization of the metro systems are planned in this direction. So, we are just talking about what sustainable resources which as a under this policy which this policy talks about, and what are the sustainable sources or resources which can be switched over for a sustainable ecosystem. So one of the very important alternative fuel or the very important resource which we can harness is biofuels. Now, what are biofuels? Any hydrocarbon fuel which is produced from organic matter, which can be a living or once living material, in a short period of time is considered as a biofuel. It is commonly advocated as a cost-effective and environmentally benign alternative to petroleum and other type of fossil fuels. As we know, the fossil fuels are continuous; they are less in amount, and they are continuously degrading at a very faster pace. So we need to switch to such sources of energy, alternative sources of energy, and one of the alternative one of the sustainable alternative resource which we can switch to is biofuel. In 2022, biofuel consumption for India was 91.72 thousand barrels per day. Biofuel consumption in India increased from 3.3 thousand barrels per day in 2003 to 91.72 thousand barrels per day in 2022, growing at an average annual rate of 26.71%. So more and more people or the government are also giving a lot of incentives in switching to these sustainable sources of energy. As we can say, there are two type of biofuels. If we classify the biofuels, they can be classified as first generation biofuels, which are also known as conventional biofuels. And what are these conventional biofuels? They are made from sugar, starch, and vegetable oils. And they are considered, such biofuels made from feedstock that can also be consumed as a human food are known as first generation biofuels. For example, ethanol, which is consumed, and gasohol. And second generation biofuels, which are also known as sustainable feedstock, they are basically they are obtained from the food crop through certain food products. Those certain food products can become second generation fuels when they are no longer used for consumption. So second generation biofuels are also known as advanced biofuels, and nowadays use of algae and cyanobacteria are also being explored, which are therefore considered as third generation of biofuels, which are holding a lot of promise, and more research is being done in this particular area and to explore their benefits. They yield about 10 to 100 times more fuel per unit area as compared to the second generation biofuels, which were considered more advanced versions of the biofuels. What are the advantages? There are some of the advantages which are associated with biofuels. They are cleaner fuels. They are more efficient. They gives more fuel efficiency. Another important thing is biodiesel, which is another very good example of biofuel, which has which emits fewer and lesser polycyclic aromatic hydrocarbons, which are sometimes linked to cancer. So they are less polluting also. And more importantly, they are biodegradable, which is the need of the hour nowadays. Now, there are certain challenges also associated on switching to these biofuels or alternate sources of energy. Some of the challenges are, first and foremost, the challenge associated is food security. The food security challenge for India has increased, including major crops like corn, rice, and sugar to produce more biofuels. So more and more farmers are switching and producing these crops for generation of the biofuels, and this is giving a negative impact on India's food security. Second important challenge is promoting and incentivizing the biofuel crops will lead to deforestation and decrease the production of other crops. Another very important challenge is it is less cost effective research and development in producing biofuel and blending technology. There's a need to finance research and development of advanced third and fourth generation biofuels as well. So more and more conventional biofuels or second generation biofuel research is being going on, but no more incentives or no more focus is being given to the generation of the third generation or fourth generation biofuels, which is also need of the hour, and more research has to be done. Now, what is the need for development of alternate biofuel crops? So it is basically to produce 60% more food on the same amount of land, make farms, field, and landscape more resistant to climate change, massively reduce the greenhouse gas emissions from the land use, improve the food security, and last but not the least, ensure bioresources and non-renewable sources of energy sustainably by utilizing lesser fossil fuels and switching to alternative sources of energy in terms of biofuels. So some of the examples which I'm going to discuss during the presentation are, which are very promising alternative sources of energy. One of the alternate source of energy, which is very much, it is conventional also and also very much in use, is biogas. It refers to a gas produced by digestion of anaerobic bacteria of the biodegradable material, such as manure, sewage, municipal waste, green waste, plant material, and crops. It is a renewable energy source. And furthermore, biogas can be produced from regionally available raw materials such as recycled waste. So recycling of the waste is also need of the hour, and more biogas can be produced using this particular type of waste. There are some of the benefits which are associated by using biogas as an alternative fuel. One of the benefit is manure, which is left to decompose, releases two main gases, that is nitrogen dioxide and methane. Nitrogen dioxide warms the atmosphere 310 times more than the carbon dioxide. And similarly, methane 21 times more than the carbon dioxide. Thus, by converting the cow manure into methane biogas, the million of cows will be able to produce 100 billion kilowatt hours of electricity, enough to power the millions of homes across the country. And owing to its simplicity in implementation and use of cheap raw materials in the villages, it is one of the most promising environmentally sound energy sources for rural needs. Second important alternate source of energy is biomass. Biomass is a biological material derived from living or dead organisms. Many biomass fuels generate lower levels of atmospheric pollutants in terms of sulfur dioxide, which are main contributor towards acid rain and lead to degradation of it leads to degradation of the environment, lead to lot of diseases when it falls on the earth, because it leads to skin diseases, cancerous diseases, someone who happens to come in contact with the acid rain. So biomass residues, co-products and waste which are not used for the generation of energy will lead to production of carbon dioxide, methane, which will eventually contribute to the environmental pollution. The use of biomass fuel provides an economic incentive to manage woodland, which further helps in improving the biodiversity. So biodiversity is very important for our ecosystem. It has to be preserved, and therefore biomass fuel is also one of the promising alternate source of energy. Another important alternative fuel here is ethanol. Cellulosic ethanol is commonly made from sugarcane, bagasse, or from various grasses that can be cultivated on low quality land. Cellulosic ethanol is predominantly used as a gasoline additive and as the primary source of biofuel in North America. The National Corn to Ethanol Research Center is a research division of South Illinois University in US and is dedicated solely to ethanol-based biofuel research projects. But now in India also, a lot of focus is being given for the utilization of ethanol as another alternate source of biofuel. So Brazil, if I talk about the advantages of this particular alternate fuel, ethanol, is that Brazil stands first in terms of bioethanol consumption in the world, and carbon monoxide emissions from automobile decreased from 50 gram per kilometer in 2015 to 5.8 gram per kilometer in 2021 by switching to this alternate source of energy. USA and Australia use 10% ethanol blend.
Ma'am, last 1 minute.
Yes, sir. and switching to alternative fuel, and they are more and more switching to alternate sources of energy gradually. India is the fourth largest producer, and the government mandated the use of 55% ethanol blend in petrol to be sold in 9 sugarcane producing states. So it has become, the government has given this mandate that India should use 5% ethanol blend in petrol in some of the states which are producing high amount of sugarcane. So algae and cyanobacteria, another very important source. The use of algae and cyanobacteria as a source of third generation biodiesel holds promise, but has been difficult to develop economically. In addition to this projected high yield, alga culture, unlike crop-based biofuel, does not entail a decrease in food production, since it requires neither farmland nor fresh water. Many companies are pursuing algal bioreactors for various purposes, including scaling up biofuels production to commercial levels. So in conclusion, for India to achieve resilient and inclusive cities, it is necessary to continuously plan for low carbon model of growth in our cities rather than focusing on physical infrastructure for vehicle mobility alone. The need is to switch over to low carbon generating biofuels as an alternative source of energy that are also less polluting for the environment and less degrading in nature. Thank you so much.
Good afternoon, everyone. First of all, I'm very thankful to the organizing committee for giving me this opportunity to present some of the work here in this conference. I'm Dr. Pratibha Kumari, Associate Professor at Deshbandhu College, University of Delhi. And my research group is working in the area of bioorganic materials. We are also working on environmental remediation, catalysis, green synthesis, and some medicinal chemistry field. So today I'll talk about extraction of cellulose from agro-waste and its utilization in developing active food packaging films. So my slide is not changing.
Yes, ma'am, it is not changing.
I don't know.
Ma'am, could you please unshare and then share once again with the entire screen? Not only single tap, entire screen, ma'am.
Okay.
Just try now, ma'am, if you're able to change the slide.
Now I'm changing slide, but it's not in the presentation mode. Is it fine?
Yeah, it's fine, but we are not able to see the slide change, ma'am. Could you please go to the next slide?
Okay.
No, it's
But
still on
still
the.
it is not changing.
Yeah. Are you sharing the single tab or sharing the entire? Yeah, now you insert. Yeah, now it is changing, but it's not in the presentation mode. That is fine, but it is changing now.
Okay, now it's changing.
Yes, ma'am.
Okay. Yeah. Now I'm talking about the food. The food that we consume is badly affected by the environment, because if the environment is contaminated, our food get contaminated. Because in the environment we have various kind of pollutants, and some of them are highly persistent. So their persistence in the environments create a lot of problem, because we human beings are being exposed to these harmful chemicals, and that's why we create these harmful chemical causes different kind of health hazards to human beings. So that this environmental pollutant has become a global issue. So we have to work in the direction of these environmental pollutants, that how can we remove these pollutants from the environment, and how can we look for the some bio based material, so that we can avoid the use of harmful chemicals. So there are different sources of harmful chemicals in the environment. Now today I'll talk about one of that is food packaging materials. For food packaging, different type of materials are being used, and after consuming food, generally we discard the packaging material, and which creates various kinds of bad effect, or it enter into our environments and creating health hazards to human beings. So most commonly used packaging material is plastic. So we pack all the foodstuffs in plastic, even water is packed in plastic. So we generate different type of plastic waste. So this plastic waste we dispose in the land or on water bodies. So through the land and water bodies, they generally enter to our in the soil, and then through the soil they are present in the food stuff that we are getting from the plants material, and even in the fish and sea, these harmful chemicals are there, and in the drinking water also it is there. So we are consuming these harmful chemical, and we know about the microplastic. It is there due to presence of the plastic material, because plastic is the most commonly used packaging material, and this plastic is creating various health problems like liver toxicity, digestive toxicity, reproductive toxicity. People are facing immune problem, and even there is a risk of cancer by consuming these bioplastics. So what could be the alternative of this bioplastic? So the biopolymer can give a alternative to these plastic materials, and we can easily get these biopolymer from the plant sources. So plant sources can be used to extract biopolymers. Biopolymers are biodegradable. So in this way, we can get some renewable sources to get the material which can be used for the packaging material. So they are the different type of biopolymer which can be used for packaging. But today I'll talk about the most versatile biomolecule, that is cellulose. And we know the chemistry of the cellulose. The first time it was discovered by a French chemist, A. Payen, in 1838. So it's not a new compound. And its first polymeric structure was established by Studinger in 1920. And it was synthesized in lab also by the Kobayashi and Soda in 1992. And we know it is the most abundant polymer, and it is present in the cell wall. So we can easily isolate it also from the natural sources. And we know in the wood it is about 40 to 50%. And in cotton, there is 90% cellulose. So these are the very common raw material from which we can easily isolate cellulose material. But nowadays, a lot of focus is on conversion of cellulose to nanocellulose, because nanocellulose is more versatile. It has high surface area, its mechanical properties are superior, and it has high hydrophilicity, its strength is high. So that's why now most of the researchers are working in the direction of nanocellulose application. And we can easily prepare nanocellulose from the cellulose. We can easily hydrolyze the cellulose into nanocellulose. Even we can do the oxidation of cellulose to produce nanocellulose. So it is very easy to prepare nanocellulose, and nanocellulose has various applications. And according to a study, the nanocellulose market is expected to grow from 0.4 billion in USD in 2022 to 2 billion USD by 2030. From this data, we can see the growing market of the nanocellulose and the growing application of the nanocellulose. Nanocellulose has various applications. It's like it can self-assemble into liquid crystals, so it can show that type of properties. And even we can prepare different kind of hydrogels, aerogels using nanocellulose. So due to various kind of properties and application of this nanocellulose, now nanocellulose is being used in tablet coating. It is also being used in drug delivery system. Even the biofuel production, like we can use nanocellulose and we can convert it into biofuel. That is also a growing area. And nanocellulose is also used in frozen milk drinks and even in the ice creams. And the UV blocking film can be prepared by using nanocellulose because it has a property to block the UV radiation. And it can also be added to cement to reduce shrinkage during drying. And it also increases sound absorption. So these are the few application area of the nanocellulose. And now we'll talk about how can we isolate nanocellulose. We can use agriculture waste to isolate nanocellulose. We know cellulose is present in the cell wall. So we can take any kind of agriculture waste, like we can use wheat straw, we can use rice hulls, we can use bagasse, and any agriculture waste which we can use to isolate the nanocellulose. So as in the cell wall, along the cellulose, there is hemicellulose and lignin. So the task is to remove hemicellulose and lignin. So by some physiochemical treatment, we can easily remove lignin and hemicellulose. We'll get this type of fragments of the cellulose. Then by further chemical treatment or delignification and by the functionalization, we can produce different form of the nanocellulose. Like we can produce nanocellulose in spherical form. We can also produce nanocellulose in the fibrous form. Even we can produce them in the roll-like structure. So we can prepare different forms of the nanocellulose based on the treatment method and the sources. Like in this table, we can see if we change the method and the source, we can prepare different type of nanocellulose. Like if we take the cotton as a source, in the cotton there is 80 to 98% cellulose. So if we apply acid hydrolysis process to convert cellulose into nanocellulose, then we'll get CNC. CNC is the cellulose nanocrystals. So we'll get the crystals of the cellulose. And here you can see the morphology is different. We are getting 177 nanometer long structure and 12 nanometer wide structure. If we use rice husk for extraction of cellulose and conversion to nanocellulose, there is 35% cellulose in rice husk. So here we can apply alkali bleaching treatment and the tamper oxidation process. Then we'll get the nanocellulose fibrous structure. And here average weight is 5 nanometer. And we have the length in micrometers. So depending on the source that we are using, the agriculture base that we are using, and depending on the chemical treatment method, we can prepare different types of nanocellulose materials. And these have the different application and properties. Now here we used wheat straw to isolate nanocellulose. So first of all, we wash the wheat straw and we grind it. Then we treated it with alkali. We treated it with 2% NaOH. We heated it, and this leads to partial delignification because the lignin is there. So some part of the lignin gets removed when we treat it with the alkali. We'll get the raw cellulose. Then we performed bleaching by using acetic acid and perchlorate iron. And after we repeated bleaching 3 times, after bleaching these hemicellulose and the there are some crystalline portion in cellulose and there are some amorphous regions. So amorphous region get hydrolyzed or it get removed by the bleaching. We'll get the purified cellulose. Then we perform the acid hydrolysis. So after acid hydrolysis, we'll get the small fragments of the cellulose. To make it further refined and to make it further smaller in size, we perform tamper oxidation. After oxidation, we'll get the fibrous structure of the cellulose. So in this figure, we can see different stages of the cellulose. Like this is the wheat straw. When we grind it, it looks like this. Alkali treatment. Then we perform the bleaching. We dried it, and then in the last there is the tamper oxidized cellulose. So we'll get the cellulose like this. And from the TAM image and from the SAM images, you can see that the nanocellulose is having fibrous type of structure. So we isolated and we prepared this nanocellulose material. And then we use this nanocellulose material to make packaging material. To have a better quality or to have an active film of this nanocellulose material, we also added copper gallic acid metal organic framework to produce anti-oxidizing property in the film to make the foodstuff safe. So we prepared the copper gallic acid MOF metal organic framework. In the figure, you can see the scheme. Like we prepared the micelle using STS. Then we added copper acetate and gallic acid into this solution. We stirred it at 70 degrees Celsius. This leads to the formation of metal organic framework. Then we added ethanol to get the precipitate of the MOF. So we prepared this MOF and we added this MOF in the nanocellulose film. But nanocellulose is does not make a very good quality film. So to have a better material to make a film which is quite stable, we use chitosan also, because chitosan is a film forming material. So we use chitosan as a base material, and then we mix nanocellulose and the gallic acid MOF into it. And then we prepare different type of films. Here you can see we prepared, this is the film of the chitosan. There's nothing is added, just the chitosan film is there. Then we added 5% nanocellulose in the chitosan, we'll get this type of film. 10% nanocellulose, this is the film. Then we added MOF in it, 1.5% MOF is added, then 2.5% MOF is added. And in this film, you can see there is a combination of nanocellulose and the MOF. And then we increase the amount of the MOF. Then here we increase the amount of the nanocellulose to 10%. Then again, we increase this amount of the MOF from 1.5% to 2.5%. So these are the different combinations. So we use different combination of nanocellulose and the mauve in the chitosan film. Because chitosan is a very good film forming material, so we added nanocellulose and mauve in this chitosan film. Then we studied the properties of all these films. So first of all, you can see the TTA graph of all these films. These films are not stable at high temperature, but at room temperature or at the temperature less than 100 degrees Celsius, we can use these films for packaging application. These are quite stable at room temperature, so we can use for the packaging at room temperature. And in the next figure, we are showing the UV visible spectrum of the all synthesized film. Like our focus is only the UV radiation. So we can see here, when we added nanocellulose, this is the chitosan film. The black one is the chitosan films, because chitosan is not showing any effect. Like it is not, it is transmitting the UV light, and it is not, it is badly affected by the UV light. That means in the UV visible range, you can say the UV range mainly, the chitosan film is showing the transmittance. But when we add mauve and nanocellulose, it is not showing anything. That means it is it is not transmitting the, or you can say it is not transmitting the UV light. So food can remain stable. It may not be affected by the UV light. So the combination of nanocellulose mauve, when we use 5% nanocellulose and 1.5 mole % mauve, that's the best combination, which is showing protection from the UV radiation. Then some other properties were also studied, like the moisture content. So moisture content, you can see here, we added different nanocellulose percentage. Here there is no nanocellulose. There is 5% nanocellulose with different ratio of the mauve. So with 5% nanocellulose and 1.5 mole % mauve, you can see here moisture content is less. So that means it is not affected by the moisture. So it is the good combination where the moisture content is not affecting the food stuff. Then in other property is the water solubility. But water solubility, again with different weight percentage of the nanocellulose. Here water solubility is least and there is no nanocellulose, but with 1.5% mauve, it's high, but still its other properties are good because its moisture content property is good. So that's why we preferred this 1.5% of mauve. Then this is the swelling degree. You can see here swelling degree is almost same in all the cases when we add 1.5% mauve. So we focused on other properties then. And this is the peroxidase value property. That means the effect of oxygen. Okay, so peroxidase value is least when we have 5% nanocellulose and 1.5% mauve. So it is least. That means it is not affecting, the oxygen is not affecting the, it's not passing through the this nanocellulose film. So it is not affecting the food stuff. So all these properties favors that when we have a combination of nanocellulose and the mauve, it is having better antioxidant property and it is not getting affected by the moisture and the oxygen. So food remain quite stable. And we also performed the real packaging studies. We also studied the, its tensile strength, and and we monitored that the food stuff was stable for a long duration of time when we use this type of film. But we are not showing all the results here. So that's why we can say we synthesize a very good film to protect the foodstuffs. So in the last, we can conclude that the nanocellulose is a nanofibrous material that has very good mechanical strength, and it also has flexibility. And we can easily make it transparent and lightweight packaging material by using this. And it has exceptional barrier properties because it acts as a barrier to the moisture, it acts as a barrier to the oxygen, and it blocks the UV radiation also. So we can easily use it for packaging different type of food material, and we can extend the shelf life of the food products. And in the last, I acknowledge DST served UGC and DRDO for the research funding, and Deshmukh College, University of Delhi, and Department of Chemistry of University of Delhi for the research facilities. Thank you so much.
Good afternoon to all. Myself is Dan Bahadur Pal. I am Assistant Professor in Department Chemical Engineering, SVTU, Kanpur. Now I am presenting the sustainable removal of arsenic from simulated wastewater using waste biomass seed pod bioadsorbent. The content of the slide is introduction, material method, result discussion, conclusion. So first we see the introduction. We here we choose the amaltas because in the area of the West Bengal and Jharkhand, this is the abundantly available in the forest. So we choose here the amaltas pods, also called the golden sour tree. The scientific name is Cassia fistula. It the blessed with the myriad health benefits as well as different important applications such as biochar, essential bio ingredient. It is widely used for treating flu, feverish condition, managing cough, cold, sore throat, threatening digestive, and different application. But we have used here for the bioadsorbent as a biochar for removal of arsenic. So basically we have choose the pods of amaltas and collected in the local forest in near to BIT Mesra because earlier I was there and then I have and our colleague also with these two we have worked these work and other that will be the firstly we collect the samples from the forest and clean wash and then pulverize it and then we have calcine for the biochar for application of adsorption for synthetic wastewater. This is the flow chart of the particular work for preparation of biochar. First, dry the amalthas peel and seeds, then crush and mild, then powder after sieving. Then we have to before calcination and after calcination we have characterize it for the elemental analysis, morphology, a functional group, and crystal size and crystal structure. Then calcine at 400 for 3 hours for making it at a biochar for bioadsorbent application. So first we have the morphology check here before the adsorption and after adsorption. We see before the absorption is the nature like lump, and after the absorption, then the structure of morphology of the particular sample will be changed. Next, we have characterized through the functional group. This is the also before absorption and after absorption. Blue one is the before absorption and red one is after absorption. There will be the some functional group will be changed the location during the adsorption of arsenic 3. Then we characterize it through the crystal size and crystalline structure for the XRD. And we know that the biomass, bioadsorbents has, there will be like a calcium, magnesium, and these oxide will be formed after the calcination. So here we have seen as the different 2 theta value. There will be the calcium quartz, cellulite component. In other scientists also referring the same. Then we have see the mechanism, adsorption of the adsorption kinetics of particular. We generally the given the taken the 3 or 4 components. It means concentration, different pH, and different adsorption dose. It means all experiment done around 60 minutes they give the 75% adsorption of arsenic was done, but after 80 minutes that will be increased up to 91. In that, the adsorption kinetics, we have to see the first order and second order kinetics, also yellowish and interparticle diffusion, but best fitted here is the pseudo first order in the arsenic 3 removal. Next, we go for the adsorption isotherm. Here also, we have chosen three models, Langmuir, Friendly, Temkin, but in this case, the Friendly model will be suited because R square value is higher. Then we have seen the effect of dose, pH, concentration of the particular adsorption of the arsenic. So here we see the effect of dose and pH of particular removal. In that case, the dose will be optimized. In 60 milligram dose given the best result, and it means best removal for particular arsenic 3. Similarly, in the case of pH will be 6. At 6 pH, it means acidic region that the removal will be better. Here the also we see the optimization, similar effect of dose, pH and concentration that will be same here. Now we see the adsorption capacity at different concentration of arsenic. So if we see the maximum adsorption capacity given the at a concentration 2 ppm, that will be maximum adsorption 60 milligram dose, and pH at pH 6, that will be give the better adsorption capacity as well as the removal. So conclusion, it was noticed that the biomass bioadsorbent potential to remove the arsenic contamination from the simulated wastewater solution for removal around 91% during the 80 minutes of batch experiment. Using that adsorption dose, it means dose will be 60 milligram, pH 6 at temperature 30 degrees. It means temperature will be room temperature around 30 degrees that time. The arsenic adsorption behavior also we see the kinetic models and isotherms, also the first order kinetics follow and friendly adsorption. So thank you. This is the regarding the.
Can you proceed?
Good afternoon to all. So thank you for this opportunity. So myself, Dr. S. Saravanan, Associate Professor in the Department of Physics, Swarandhara College of Engineering Technology, Narsapur, Andhra Pradesh. So today my topic, surface morphology influenced in barium sulfate nanostructures by solvents with Azadirachta indica leaf extract. So this is my overview of my presentations. First, I'm talking about motivation, next experimental approach, resultant discussions and conclusions, finally references, what I have used for this. So first, motivation. So why I am involved in the green synthesis. So the green synthesis based nanomaterials are environmental friendly, which is viable and better alternative method for various applications. In that, I have selected barium sulfate material, which is unique structural, electronic, optical, and chemical properties because of their size changes from bulk to nano scale level. It is widely used for many applications like shading material for the X-ray photography, gamma ray absorber, white pigment, etc. However, my target is to utilize in the antimicrobial activities. So this is my experimental approach. Here you can see the diagrams. First, it is showing that the neem leaves, that is scientifically we call Azadirachta indica leaves extract that we have taken from water. It was boiled at 80 degrees Celsius for 1 hour. Then we got that extract, then added into the water. Then barium chloride and sodium sulfate, both are primary to get barium sulfate. So finally, I got white precipitate. So again, I kept in the drying process with 100 degrees Celsius, 2 hours. So finally, I got the same morphology. You can see that in the last image, that is ethanol and DI water. It means that the solvent I have used first water and ethanol. So both materials, both solvent I used to compare their morphology and growth. So first you can see the diagram. This is ethanol-based morphological structures. This is DI water-based morphological structures. So here I have prepared two different samples. Result and discussions. After preparations, I went for few characterizations techniques to compare their optical, structural, functional, and morphological structures. You can see that first XRD, next FTIR, scanning electron microscope, and last EDX. So these are all the characterizations technique what I have carried out. So first you can see that X-ray diffractions. This first it is showing that ethanol based XRD. Next is showing that DI water based XRD. You can see that structural properties. DI water shows more intense and sharp diffraction peaks. However, ethanol show little broader diffraction peaks, and then the peaks around 24.39, 28.9. 32.3, 46.7. These are all well matched with the JSPDS files. So it is confirming that barium sulfate is presenting. Next, FTIR. Here you can see that first FTIR spectra representing ethanol-based transmittant spectra, and then second C representing the DI water-based FTIR spectra. Here also you can see that the sharp peaks appeared in the water-based FTIR spectra. And then here the 1200, 1300, these are all confirming that sulfate is presenting. However, the 1735, 1726, these are representing the barium is presenting. However, that 2000, around 2913 and 3000, these are representing the water molecules are presenting. Here, this is scanning electron microscope. You can see that the first two representing ethanol-based morphological structure, and second DI water-based morphological structures. You can see that the first two representing the smallest nanostructures. This look like the rod, but the average, when we are taking the average of that size, it is coming around 77 nanometer. However, you can see the second, the below two diagrams representing that is water-based nanostructures. It is clearly visible that the agglomerations are very less and it is free. As well as you can see that it is look like nanorods, and the average we got around 225 nanometers. Here, not only I'm changing the solvents as well as the molar ratio, also I changed the ethanol. I have used 0.05 molarity, and then DI water I have changed 0.1 molarity concentrations. So these are the differences, how the growth are there. And last, the energy differences, energy dispersive X-ray spectroscopy. This is useful to know the element are presenting or not. You can see that figure A representing ethanol-based nanostructure shows that barium, sulfur, oxygen, and then sodium, these are all elements are presenting. Here some more elements came because of the calcination, and then that is substrate, whatever they are using during the EDX. So few impurities are came. However, you can see that the majority, barium 43.69% and sulfur 12.12%, and then oxygen 22.5%. These are all ethanol-based nanostructures and their element. Again, figure B, you can see that this is deionized water-based nanostructures. You can see that here also barium and sulfur, oxygen are presenting with impurities. However, the weight percentage are less as compared to the ethanol. You can see that barium 32.92, sulfur 9.84, and then oxygen 28.86. So these are the percentage. However, the best structures we got from DI water-based barium sulfate. Final conclusions in this work: successfully green synthesized barium sulfate nanorod structures by using different solvent and different molar ratio, molar concentrations. Here I have used a co-precipitation method for the preparations of nanoparticles. With that, the neem leaves extract I have used, that is used as a stabilizing agent. And then XRD confirming that the barium sulfate structural properties are there, that is representing the orthorhombic phase. And then FTIR also confirming that barium and sulfate group molecules are presenting with the transmittant spectra. Again, the same result shows that the nanostructures are presenting, which is look like the nanorod structures, and the morphology are improved as compared to ethanol to DI water-based barium sulfate. So these successful preparations of nanostructures we got from 0.1 molarity based barium sulfate, and then EDX spectra also confirmed the element are presenting. So future, these molar concentrations, solvents would be optimized for well-defined nanostructures for antimicrobial applications. So these are the references what I have used. So thank you.